Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please take a moment to like, subscribe, and share this with your friends and family. Your support really matters, and it helps the Science Experience keep bringing you this kind of cutting-edge research. Okay, so today we're tackling something subtle but really fundamental, the invisible force behind biological aging, chronic inflammation. We've got this fascinating new paper that uses, believe it or not, the common fruit fly, and it's trying to move us beyond just, observing aging into actually quantifying it. So the goal wasn't just watching flies get old, but finding reliable, repeatable genetic markers for that silent state we call inflammaging.
That's absolutely the key shift here. We're moving past just general biological signs to creating a real metric. The aha moment in this study, I think, is developing this precise, measurable biological clock. Think of it like a genetic stopwatch for the fruit fly. They actually call it DiAge, which stands for Drosophila inflammatory age. And this new model, it's designed to show us pretty quickly and accurately how things like diet or genetics or maybe some intervention speeds up or slows down biological aging. Crucially, this is kind of independent of just how long the animal lives overall. So maybe let's start by defining our target. What is inflammaging? It's not like, the acute inflammation from a cut or something. It's this low-grade, persistent increase in the expression of innate immune genes. And this happens even when there's no clear infection present. The system is sort of stuck on high alert.
Right, so the body is constantly geared up for a fight that isn't really happening. And I guess that low-level readiness must be incredibly costly over time for the organism.
Exactly. It's costly. This persistent state of alarm, it's implicated in nearly every major age-related disease you can think of. Arthritis, neurodegenerative disorders, various cancers. So if we can measure this chronic immune hyperactivity precisely, maybe we can find ways to, modulate the aging process itself.
And to try and crack this code, researchers turned to the fruit fly, Drosophila melanogaster. We often mention fruit flies, but for aging research, they're especially powerful, aren't they? They share some fundamental immune pathways with us mammals, but their lifespan is super short, like 70 days max.
Yeah, around 50 to 70 days, typically. It lets researchers basically compress decades of human aging studies into just a couple of months in the lab. It's a huge advantage. And what gives us confidence that these findings might translate is how conserved the basic mechanisms seem to be. For instance, they noted that a human protein, alpha antitrypsin, or HAAT, it actually suppresses inflammaging in both human cells and in the flies. That suggests we're likely looking at the same core biological driver.
Okay, and the method they use to track this, it sounds pretty clever. This AG index, age-associated gene expression, is basically a ratio.
Exactly, a simple ratio. You take a gene's expression level when the fly is old, say 30 days or more, and divide it by its level when it was young, like 3 to 10 days old. By looking at this change ratio instead of just the absolute level, the idea was to filter out some of the noise. Noise from different lab environments, maybe slight variations in diet, even different sequencing tech used over the years.
That seems like a smart way to handle data collected over a long time from different places. Did it work?
It seems so, yeah. What's pretty remarkable is the confirmation it gave. They pulled together RNA sequencing data spanning over a decade from different fly strains, different labs around the world. And despite all that variability, they still found this core group of genes that showed really strong, consistent, age-associated expression changes. That consistency across such diverse data sets, it really validates that these aren't just random fluctuations. They're genuine markers.
Okay, and were the usual suspects there? The immune genes?
Oh, yeah, definitely. Confirming previous work, the innate immune genes, specifically the anti-microbial peptides AMPs, they were highly overexpressed with age. Things like the Attacin genes, Attacin, ATB, ATC, that wasn't a huge surprise.
But there was a novel finding, right? Something that really stood out.
Yes, exactly. While the AMPs were expected, the team identified one particular gene that gave an exceptionally high signal. It's called SPH93. That stands for serine protease homolog 93.
And I read that SPH93's signal, its AG index, was so dramatically high, it basically threw off the charts. They sometimes had to leave it out of plots just so you could see what the other genes were doing. That sounds like a massive effect.
An undeniable marker in terms of correlation, absolutely. And its human equivalent, or ortholog, TPS-AB1, that's already linked to inflammatory issues like asthma in humans. So that adds weight. But, and this is where science gets interesting, right? There's a catch. Sequence analysis suggests SPH93 might not actually have a functional protease part, the bit that usually does the cutting work.
Wait, so it's a massive signal for aging? Its human version is linked to inflammation. But the fly gene itself might not be functional in the way its name suggests. That sounds like a pretty big functional mystery. How does that affect things, like building the DiAge model?
It certainly makes figuring out its actual function a top priority for future research. It's a huge question mark. It tells us the mechanism driving this specific part of inflammaging might be, well, different from what we'd assume based on the name. Maybe it acts structurally. Or perhaps its role is purely regulatory somehow, kicking off other pathways we don't know yet. But regardless, its super strong correlation makes it a vital piece of data for the model, even with the functional uncertainty.
Okay, so the reliability of these core markers is strong, but the study also showed it's not that simple, right? The organism itself adds complexity, like differences between males and females.
Exactly. Significant sexual dimorphism. They found that inflammaging patterns are often, let's say, female skewed. Certain immune defense genes just had a much higher AG index, a bigger increase with age, in females compared to males.
And why is that difference so critical to track? You mentioned confounding factors.
Yeah, it's a great example of why careful experimental design matters. Take one gene they looked at, SEC1. It turns out SEC1 is highly expressed in young, perfectly healthy males, but mainly in their reproductive tract. So if you just grind up a whole body sample, that high expression in young males artificially boosts the young number in your AG index calculation. And that makes it look like there's less age-related increase in other tissues than there really is. You basically mask the true inflammaging happening in, say, the brain or the fat body.
Ah, I see. So you think you're measuring aging across the whole body, but really you're just measuring how much reproductive tissue was in the young male sample versus the old one.
Precisely. It obscures the real picture.
And it's not just sex, right? The timing is different depending on the body part, too.
Yes, absolutely. Organ-specific timing. For one fly strain they focused on, the W.O.DA strain, they saw clear signs of inflammation in the fat body, which is kind of like our adipose tissue or liver, relatively early, around day 30. But in the brain and the thorax, inflammation was pretty minimal then. It only really ramped up much later, by day 50. So different systems are definitely aging at different speeds within the same animal. Monitoring tissue specifically is complex, but clearly necessary.
Okay, so we have these reliable markers like SPH93, but they might be functionally mysterious. And we have all this complexity from sex differences and tissue-specific timing, which I guess leads us to why they needed a more sophisticated tool, this DiAge model, built with machine learning.
That's right. It's inspired by similar approaches in human studies, like Steve Horvath's work on DNA methylation age clocks. They used a technique called elastic net regression, feeding it loads of RNA-Seq profiles from flies of known ages. The goal was to train the model to predict the fly's inflammatory age based purely on its gene expression pattern.
And how accurate did it get? You mentioned it performed well, especially for males.
Yeah, when they focused the model just on male samples, they got the mean absolute error, the MAE, down to about 1.57 days, which is pretty precise for a fly lifespan. And the model revealed some interesting patterns. Genes that correlated positively with this DiAge, the ones driving the inflammatory age up, were strongly linked to the antifungal humoral response, part of innate immunity.
Antifungal specifically, not just general immunity.
Yeah, that was the strongest signal, yeah, the antifungal response. And conversely, the genes that correlated negatively with DiAge, the ones whose expression went down as inflammatory age went up, were involved in mitochondrial energy metabolism.
Okay, that combination, mitochondrial decline alongside immune ramp up, that sounds like a classic hallmark of aging seen in mammals too. But the focus on the antifungal response as the top predictor, that's intriguing. Any idea why that specific pathway?
Well, it suggests the DiAge model isn't just picking up general immune noise. It might be highlighting a specific kind of preparedness signal. Maybe it's not about fighting an active fungal infection, but a sign the system is constantly primed, over-prepared for a broad range of potential threats, including fungi. And maintaining that hypervigilant state is metabolically expensive, right? Which lines up perfectly with the drop in mitochondrial energy genes. It looks like a trade-off.
Right. Hypervigilance costing you energy efficiency. That makes sense. So the real test is applying this DiAge tool. Did they use it to predict anything?
They did. They applied the model to external data from flies fed a high-fat diet specifically, 20% coconut oil. And the DiAge model predicted a significant acceleration of inflammaging on this diet.
Ah, okay. High-fat diet speeds up inflammatory aging, but wasn't there a twist?
Yes, the crucial twist again. This acceleration was only seen in the females. The model detected no significant acceleration in males on the same diet. This really nails the sex-specific responses, which lines up with other studies on high-fat diets in different animals, including potentially humans.
So the DiAge model isn't just a score. It's a predictive tool that can reveal these kinds of biological nuances. That seems incredibly valuable.
Absolutely. Its value is potentially huge. Instead of running these long, cumbersome lifespan studies that take months, researchers could, in theory, test an anti-aging drug or a diet change and measure its impact on biological age acceleration much faster, maybe at any time point they choose.
And the accuracy seems pretty good for that. The error rate was about 3.35 days overall.
Yeah, the median absolute error for the combined model was around 3.35 days, and the researchers did a rough calculation, suggesting that's maybe equivalent to less than 4 human years. That kind of precision could really speed up the process of finding effective interventions.
Okay, let's try and wrap this up then.
Sure, so the big takeaways, I think, are first, finding this core group of really reliable inflammaging markers, like that standout SPH93, which hold up across different fly strains in labs. That's foundational. And second, the development of this DiAge machine learning model. It gives researchers a powerful new tool, a validated method to actually measure the speed of biological aging in the lab, not just its endpoint.
So thinking about the implications for us, for you listening, since that DiAge model clearly showed this significant sex-specific speed-up of inflammatory age in female flies on a high-fat diet, and given that connection between inflammation and mitochondrial decline, what might that finding suggest about human dietary guidelines? Especially, could there be a need for sex-specific advice as we get older? That's definitely something to think about. Thank you for joining us for this deep dive. If you enjoyed getting well-informed quickly, please do remember to like, subscribe, and share it around. Your support genuinely helps the Sand Science Experience bring you more of this cutting-edge research. We'll see you next time.